Welcome to this M2D2 talk. Our speaker today is Professor Gianni de Fabritiis, and his talk will be titled Machine Learning Molecules. Professor Gianni is from the University of Pompeu Fabra in Barcelona. He leads the computational science research group dedicated to computational science and in biomedicine and machine learning, and the distributed computing project GPUGrid.net. He's also the founder and scientific advisor of the technological company named Accelera, that is focused on the use of GPU in biotechnology and developing biomedical simulation methods. Thank you so much, Professor Gianni, to share your work with us here today. And without further ado, we can start.
Thank you. Thank you so much for the invitation. So how do I get rid of this? Here. Okay, so the title is Machine Learning Molecules. I'll have two parts. The first part, the first half, can do a little bit more of a summary of what we've been working in the past since we started to working on this in 2017, and then a little bit where we're going or where we'd like to go in the second part. So, okay, so the first, what we're trying to do, so we would like to give solution to the biological problems, so to biology, but we do that from physics or fundamental perspective. And the way to do that is like you can, so everything that here on the right, can you see my? No.
Yes, but you can use the red laser if you want to.
Or is it? Okay, well, if you see the mouse is fine. Then, so here on the right, you have the extreme complexity that you have this inside a cell. And you can go and zoom in. These are the scale. On the right, you have scale of the micrometers and 10 to the second, 10 to the minus 3 seconds and above. And all of these little things here are actually protein complexes, and which are like 10 to the 100 nanometers and these faster time scales. Then you can go and isolate individual proteins. Here, the green part here, 1 to 10 nanometers. And then you end up at the atomic level. And the nice part here is that where you have 1 to 10 Armstrong and 10 to the minus 9 seconds is the typical time scales. The nice part here is that we actually have a fundamental description of this part on the left. So in theory, we could go all the way to the right. It's just a computational problem. But of course, it's impossible because you see here the complexity that we have. So the question here that I'm asking is machine learning the right language for this sort of complexity? And I think it is for the reason that say here, there are fundamental descriptions of nature here, which is exact in terms of quantum mechanics. There are also at every level, there are kind of experiments that one can do, so you have data. But practically on the left, you have infinite data. So you can generate as much data as you like by just running more quantum simulations. So it's kind of self-consistent. You can generate more data quantum mechanically and then kind of extrapolate machine learning model on the right, which is what we want to do. It's impossible to tend to simulate the cell at the level of quantum mechanics, of course. Okay, so what have we done to do this? So once AlexNet came out, a few years later, we started to see if we could apply deep learning to molecular structure. This was back in the day when there was actually only us and another group working on this, Atomwise and us. And the way that we approached the problem back then, there was, okay, if images are matrices are like stacked matrices, we can describe protein as stacked vector tensor or three-dimensional tensor. And one of each color could be some sort of descriptor of the protein. This is the bottom here is a protein. So it could be atom properties of anything like this. And back then they were not. So when we started to do this, just 3D convolution came along in Theano, so we could actually start to do something straight away. And the first kind of application of this sort was DeepSight. I put reference down here. It just, again, is kind of a deepish neural networks where we take this slightly box, so this concept of three-dimensional photos, if you want, the images of these proteins. And the idea was to be able to predict where a drug would bind. And these are these oranges part here in this picture over here. And this we were rasterizing over the structure and with our 3D image and building this thing. So this is one of the first application that you can see around of machine learning in structural biology for prediction using deep learning. And the second application that we did was a similar network. We were trying to predict binding affinity. Actually, I forgot to add the citation here. So KDeep was again a predictor that you put in a protein in a small molecules, and it would predict some sort of some affinity, so how much the two like each other. And this is the result on a PDB bind data set, how well this this model was working for given targets, for given proteins. We did again something similar, but so this one was a regressor. Here we did kind of the same, but it was a classifier. And again, same structure as before, but just trained as a classifier for deciding if useful for virtual screening in the case that we want to see if the molecule is a binder or not how much it binds. And another application that we did just slightly later, this time in collaboration, this was 2019 with Pfizer, Biogen, and Janssen, was so if you have a couple of molecules, how the two compares in terms of affinity. And again, this was done with a special architecture, which was respecting the symmetries of the two pairs. So you could invert the pair and the results would be the same. And we applied this kind of tools. I think this was also 2019 in this D3R challenge. The D3R challenge is a blind challenge, which is done in the community, and practically they give you a target, in this case base cats, and they give you a certain number of small molecules, and they ask you to predict the structure and the affinity. And in this case, with these methods, KDeep in particular, we managed to win 2 of these challenges, the one in black over here. And so to say that these deep learning methods were kind of a state of the art already after 1-2 years, okay, compared to other techniques that have been available. We also worked on generative models of structures, and this seemed a very good idea. There are, I actually don't remember where I took this picture, so it's not mine, it's from a paper, but I couldn't find the paper anymore. So then there are 2 ways of the generative models. So, in the first part, we just did deep learning, classifier and regressor. Then in generative models, you can try to say, okay, let's, you generate atom by atom, the molecules, or you generate them fragment by fragment, or you have some sort of reaction-based type of generation where only certain combinations of atoms or fragments are allowed depending on the generation. So the one we worked back then were atom-based kind of things, which have a lot of benefits. So it gives a lot of exhaustiveness in chemical space, structure novelty and structure diversity, but they have some big flaw, which is they don't necessarily give valid molecules, or they don't necessarily give molecules that you can make easily. And this was a major limitation. The method on the right have less of this problem, especially the reaction based. But anyway, you are here in the middle. So the one that I'm talking about is this atom based. And the first one we did was, again, in 2019, was practically an in-painting, so a conditional probability model where we were passing a protein and we were asking the model to kind of fill the pocket down here with the properties of small molecules. And so we were generating this kind of property, aromatics, hydrogen bonds, and acceptor, for which one could guess what sort of small molecule would fit there. This is not really a generative model. This is more conditional probability, but then we use variational autoencoders as well, where again, from a seed molecule, we will kind of encoding a shape representation. And this would give, again, some sort of features inside the pocket. And then it was hard for us with this sort of representation back then to kind of isolate. I mean, there are tons of molecules that fits into this shape. So what we used back then was a captioning network to go from, again, from the shape, which could be any sort of likeness of a drug, to actually proper molecules in terms of smiles, which is a kind of a text. This was again in 2019, and then we used GANs, which back in 2019 were also quite in vogue, and of course now disappeared. And again, it was the same thing as before. We're giving a structure and we're asking to predict properties inside the pocket, but this time with a GAN instead of an autoencoder. And it gave slightly better result, and again, using a captioning network to produce molecules. All of these methods had the same problem, which ultimately led us to abandon generative models, which was the molecules were not useful. So these compounds which we generated atom by atom were not synthesizable, or were not purchasable, or were not even molecules sometimes. So, of course, most recently there are other generative models that came out. One of the most promising is diffusion models. The advantage here is that instead of going through voxel, of course, the world has evolved since in just 3 years, and now everything is atom based, so point based. So you can generate exactly the atom that you want inside. And practically this is a denoising, so you start from some data and then you add noise, and then you reverse it using a reverse SDA. And then you use this reverse SDA from going from random noise into molecules. This is not by us. This is a paper from 2022, but I think it's an interesting one. He has the same problem that we had before. So the molecules that he generates are not necessarily useful, but it generates directly the molecule. You don't need to go through the captioning network and so on and so forth. So I think it's superior. And another interesting approach is to use language model in a generative way. So this is, again, not from us, it's from AstraZeneca, and this use reaction-based. So the advantage is that actually the molecules that are generated are valid molecule. And I think it's one of the most promising approach. It mixes a little bit a language model with reinforcement learning, so not too different from ChatGPT and alike. This was, however, in 2021, so a little bit earlier. And it just optimizes the language model to generate molecules which are more fit to the target, for instance. So it doesn't have a 3D structure, it's not structure-based in a sense. The only connection to the structure is through the reinforcement, so through the reward of the reinforcement learning. However, this is really nice. So one of the things that we had started to work in reinforcement learning is that we wanted to do some sort of kind of virtual chemist or acting on drugs as an automata. And like in this case, an example, like to imagine you have this target here in pinkish, and then there is some automated procedure which goes and as an agent or multiple agent that goes and search for molecules validated through multiple tools and propose molecules. So it's kind of a sophisticated version of the generative models. And so we did this PlayMolecule AI or PlayMolecule.com website, where our idea was this is kind of the gym for drug discovery. This is kind of an example. It's still not, we're still working on it, because of the problem we had on generative models. But it's still, the idea is still valid. We also worked on improving PPO for learning from examples, because we thought we had several examples in the case of drugs. And the thing we did here is like we used importance sampling, which is widely used in molecular simulation. So we use it a lot. Of course, PPO is just an application of that. And we kind of reweight correctly to integrate examples in a natural way in an online method as PPO. So PPO is on policy, so it's not like SAC that you can have of policy trajectory, but through this reweighting procedure, you can actually extend it. This was applied in Animal Olympics case, where you have this ball, which kind of needs to go through the ramp, pass over here, and eats this food over here. This was done by presenting a single demonstration, and this is what PPO learned. This is actually what the agent sees. So it just sees of this first point of view kind of approach. And again, this was just given one demonstration, then just running this PPO plus D. It needed to move the box in order to be able to reach there. So it's kind of impressive what PPO alone can do. And again, this is PlayMolecules with all the different kind of environments and that are available there. Okay, so this is kind of was the recap up to like with 2000 and all the deep learning, generative learning. Then we started to work on something closer to what. So we want to kind of machine learning chemistry or machine learning molecules in chemistry. And so we started to work on things which are kind of a little bit more fundamental in terms of atoms and so on. And the idea is that molecular simulation has been used for a while in drug discovery, and the way that is usually used in this form, the form of molecular mechanics. So you have a model, which is like down here, an atomistic model with all these atoms, hydrogen, carbon, oxygen, and so on. And because you cannot run quantum mechanics for such a big system, so even worse for proteins, there were approximate methods, which normally called molecular mechanics, where to actually build a force field which average out the electronics degrees of freedom. So you want cause graining the electronics degrees of freedom, you forget about all the electrons, and you just move around the nuclei. And on top of that, there was some other approach, which was called more really proper cause graining, where you cause grain, for instance, an entire amino acid, which is made of many atoms, by a single atom, like by just the carbon alpha. But it's kind of the same thing. If you want, you still kind of approximate, kind of averaging out degrees of freedom, not electron ones in this case, more others, like even other heavy atoms ones. And and what you're trying to build is a force field, which depends on X, which are the coordinates of all your atoms, and some parameters. Now, in molecular mechanics, the parameters are kind of maybe hundreds to 1,000 parameters. So there is a fixed functional form, which I'll show later, and you have this 100 to 1,000 parameters, which you have been learned over 2-3 decades in order to optimize kind of macroscopic property of the model up here. And then you have some sort of solver. No? In the case of molecular mechanics, you use Newton equations, so you have a simple ODE solver. In the case of quantum mechanics, you have a PD solver, but again, we're not using quantum mechanics directly here. And this is the kind of functional forms which are available in codes like SMD of OpenMM, molecular mechanics code, which is called the Amber charm force field, and it's a closed functional form where you're trying to kind of heuristically represent that two atoms like to stay close to each other because there is an electronic cloud up here that kind of force that, but you don't see the electron, so you force that with a simple spring. And so for angles and for torsions, and then you have some more realistic terms which are kind of the van der Waals, Leonard-Jones, and Coulomb, so charge interaction. So based on all of these, kind of any kind of big molecular simulation that you see are based on this simple form, and then you learn. You're not learning in a kind of systematic way, like kind of manually learning these parameters here, k theta, kb, k phi, and so on and so forth. Okay, so first thing we did was think this 2021, we wrote an entire ND code in Torch. And the advantage of doing that is of course slow. So you take, but has some advantage. So you take your atoms according Cartesian coordinate, and then you have your classical force field, the one I showed you before. But then you can add another force field, and this force field could be in a neural network, because a neural network is kind of the optimal force field if you want. So you have a many-body term where you can capture a very high-level correlation between all the high-dimensional degrees of freedom of all your Cartesian coordinates, and ends up in a number, is differentiable. So you can, if you choose the right nonlinearity is differentiable, and so you can actually differentiate through and get forces. So let's say that you from coordinate, you predict energy, and then you differentiate, and then you get on the coordinates, and then you get forces. So these two terms at the moment coexist, no? But it's one of the first network that would do that is called SNET in terms of neural network, and there's another one which ANI1, which I'm not mentioning here. And so what TorchMD does, and is publicly available here, is that it allows you to mix and match these things. So it's a development tool, and it's end-to-end differentiable. So you can actually run some simulations and then back propagate through the simulation to optimize your parameter or optimize anything. So it offers a lot of flexibility apart from just merging the two different force field. Okay, that's just what I said. So you can have force field parameter, the standard one, but you can have also machine learning potential and mix the two. Okay, once we add these, yeah.
Can I interrupt? I
Sure.
have a question regarding the previous slide. Why do you have this other part where you kind of include the term of the Amber force field instead of just learning everything from the SNET?
One of the reasons was, let's say that you have data, so you need data to sample the neural network, and two atoms might not get in any kind of your data set, two atoms don't get close enough. So you don't get close enough. They don't like to be close because because of the repulsion here. So they have this very strong repulsion when they get close. And so you don't have data when two atoms are close. And what happened there is that the neural network just goes wild there. So if during the simulation you have some sort of the atom gets too close compared to what you've seen in your training, then the thing just can get unstable, or it can get unphysical, or anything like that. So back then, when we did this, there was a kind of understanding that machine learning force field needed to some sort of supplement in terms of physics, and it's unclear if that's the case right now. So in your situation, so what you're saying, you might be correct that we don't need this part over here anymore, and we just have a simple machine learning potential. But back then, the common approach was, oh no, we need physics, and we need to have some physics, embedding some physics in there.
Okay. One follow-up question is, you were saying at the beginning of the previous session that TorchMD is slow, and I've used TorchMD, and it's slightly slower than if you're using a force field that come from a chemoinformatics toolbox. So and even the ANI and the more recent method are also quite slow. So my question is, where do you see the opportunity to actually improve the speed of this kind of ML method, like ML force field? And do you think they will ever concurrence the classical chemoinformation force field?
So I have a slide on that later, but essentially what is slow there are two parts which are slow. There is the classical forces which are slow because although they might be computed on GPU through Torch, they are still a lot slower than any other CUDA implemented code. But as you say, we might be not so concerned about these things because maybe they even disappear at some point. And then there is the machine learning potential, which are slow. And the main reason why they are slow is because of Torch itself. You have, when you do MD, you have maybe you don't have a batch. You have a single molecules, and you're updating this, and you want to update it as fast as possible. So it becomes when you use Torch model like Gianni or TorchMD-Net or whatever, it's actually most of your time is waiting for the kernels to launch. There are lots of kernels that are launching, and normally it doesn't matter because you just load up a huge batch and the kernel launch is insignificant. But in the case of OnMD, that's not what you want. You want some you want some very fast latency. So one of the reasons why these narrative potential are slow is just latency. So a way to solve that is, which we did, is to transform the model into a CUDA graph model, and that kind of package all the kernels into a single block and reduce all the latency, which especially for a small molecule, it's a lot faster, like 5-6 times faster. I don't think I have the benchmark here, but currently we can get to 5-10 times lower than the molecular mechanics. So it's not too bad. And I think there is still margin for improvement, but we'll see. So the TorchMD-Net. So one of the thing that you need is some sort of representation. So you need to represent this. So you have Cartesian coordinates for your atoms, and you have an energy at the end. And you want a very good representation model. So there was featurized representation models with simple neural network, simple multilayer perceptron afterwards, like Gianni. There are like graph neural networks and so on and so forth. And when we started working on this, we decided to make a transformer network. And what you see here on the right is kind of the proliferation of models. So each of these symbol is a model, and it's let's say this one over here, the star, is the first Coulomb matrix model, the first one. And what you see in this plot is at the bottom, you see so this QM9 database is a database of 130,000 molecules, each with an energy with a quantum mechanical energy associated to that. And what we're seeing here is the error on the column, that you get training on X amount of data. So this star over here is training, I don't know, in 10,000, 20,000 atoms and testing on some other, sorry, 20,000 molecules and test from Q9 and testing on the others. And then, of course, the more you increase the training set, so if you train on almost all the molecules, 100,000 molecules, and you keep the other 30,000 for test, then you get this better error. Now, it's a property of statistical inference that this in a log-log plot, this looks linear. So all these models scale in a certain way. So the more you increase the data, the better you get in accuracy, but this with a certain law. So the better is the model, the better is our presentation, the lower you are in these curves. So this was the first one, and then the better model, better model, better model, better model, better model, better model. I think at the moment we are below the page down here. Okay, so it got a lot better. And there are certain physical constraints that you want to have in this model. So you want translational and rotational and permutational invariance. You want energy conservation. You want equivariance. If you rotate your molecule, the forces should rotate in the same way. So that should be exact. Back then, there were two methods, non-parametric like kernel methods and parametric like neural network. And initially, the non-parametric one were better. I think now everybody's using parametric methods or somehow based on neural networks. The training loss was very simple. It was just kind of error on the energy, so on the DFT energy or QM energy compared to the neural network energy, and also on the forces. So very simple kind of loss. And so what we introduced, this was published in iClear 2022, we introduced this first transformer model, which was state-of-the-art at the moment of publication. And is what we were mainly interested in was some good representation. So you need to produce good results, good accuracy, but more than anything, we need speed, no? Because we want to compete with molecular mechanics. And this, just to give you you mentioned ANI, this is just how it compares to ANI 1X model, compare of our equivalent transformer. No? So the error in forces in both MAE and RMSE. So you see it's like 3-4 times better on the same data. So you train on the same data. This is just changing the representation. So ANI use Fisher eyes of the multilinear perceptron, and we use this transformer model. And you can take, there are now other methods which are even better. But the point is not so much to get better here. The point is actually to get faster. And that should be a main goal. Okay, so another thing that was missing were data sets. So like this year, we published this paper where we make a lot of data sets. This was mainly through the OpenMM consortium, which I'm co-lead. So yeah, we just generated a lot of DFT data for dipeptide, for solvated amino acids, for dimers, for monomers, for small molecules, ions, and so on and so forth. And also integrating lots of variety in terms of conformations, size, and elements. So all the elements that are kind of useful in drug discovery. The data set is publicly available and was built on this functional form. And again, some more description. Another important characteristic of these data sets that it has charged molecule. So there are tons of molecule with charges, and these are kind of the distributions of all the characteristics. And one thing that was kind of surprising was that actually TorchMD-Net works out of the box for charged molecules. So we don't need to have anything special to account for charges. So ANI1X is only neutral molecules, no, with SPICE, which add charges on each molecule. The model is actually able to get that right without doing anything special. And so you can see that here, because it doesn't matter the charge, so the charged atoms, the error across board is more or less the same. So it's not depending on the charge. And this is kind of this paper here is kind of where we see we're going. So I don't think it's possible in any short term or medium term to actually replace completely the molecular mechanics, just because the speed is so much more. But it might be possible to replace some parts. So in this case, what we call neural network potential /MM, we take the normal molecular mechanics force field, we take a machine learning force field and some mixed term. So for instance, you could have an entire simulation with the protein, the water, and all these bits and pieces treated with molecular mechanics, and then but then you have also neural network potential for the small molecules, which are not so good in molecular mechanics. And this could give this you can already use now, for instance, in SMD and OpenMM, and it's a possible way to use this neural network potential right now. Okay, this is the last bit. Is how we use this machine learning in for proteins, this machine learning potential for proteins as well. And in this case, we use a very coarse representation of proteins. The idea was to capture the protein dynamics. And so like this could be an all atom system when you have like 50,000 water atoms and kind of 6,000 protein atoms, and then you need to run 100 days on a supercomputer to see something here. But if you could just reduce to a more kind of beads kind of approximation with carbon alpha, then maybe you can do that in hours or days. And so what we work here is how to do this. Essentially, it's a force field. Again, it's a force field mapping. So how to build a neural network which learns from here and builds a dynamics which is correct here. So in this paper from 2019, which was led by Cecilia Clementi, what we did these methods, which is called force matching, which has some nice characteristics. So you practically try to match the forces acting on your beads. In this case, for instance, let's say we take the carbon alpha, you compute the forces acting on the carbon alpha on the latent system, and we try to build a neural network potential just for the carbon alpha. And then we generated tons of data. In this case, our fast-folding proteins, we generated all more than 9 milliseconds of simulation data, which is a lot. I think it's one of the largest data sets that you could get for molecular simulation. And then we trained this model through force matching, and actually we could get models which would fold most, if not all, of these proteins. Here you see the yellow is the remember the gray is the crystal structure, yellow and blue. The yellow is a model which is trained only on these data sets, and the blue so the blue is the model that is trained all. The orange is the model that is trained only on these data sets, while the blue is a model that is trained on the entire data sets, and it can actually fold everything. And another interesting bit, okay, this is some example of coarse grain simulation that you see that it goes up and down, it folds, unfolds, folds in just 3 nanoseconds. And okay, this how well we do. So we kind of recover, apart from Lambert repressor, we recover the crystal structure within 2 angstrom, 3 angstrom MSD. No, apart from this one, from protein B, almost everywhere. But the most interesting part is because of the way we do the training, we actually preserve the thermodynamics of the system. So what we're actually saying is that when you run our carbon alpha model, it actually is identical to take the all atom simulations and project on the carbon alpha. So you get the same energetics, same energetics in the 2 cases. And you see here, like for instance, in NTL9 or protein G, and you have a projection of the free energy of these basins in the all atom model, and the same projection on the coarse grain simulation. And you see that there are actually minima in the same place where there are minimas. Here, 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 the shape is slightly different, but there are a lot of similarity in the minima. So this is never the case in cross-graining before, which was kind of more heuristically trained. So this is one of the first methods that I know, at least, that kind of preserve the thermodynamics of the system. So it's proper coarse graining, if you want. And we also try to see how much it generalizes outside the training set by making some mutations. This is trained only on 12 proteins, so you cannot expect that can fold 13 protein or 14 protein. Is trained on a very small subset of proteins. But we try to add mutations of this protein and see if it can still fold it, and it could still fold some of them. And okay, this is just one example of one dynamics on this carbon alpha, and now it looks realistic. So, I mean, if you've seen all atom simulations of folding, these usually work realistically well. This is another one. So it's not just collapsing into the structure, the folded structure, but it's actually having its proper natural dynamic until it gets to the, yeah.
How fast this coarse-grain simulation with TorchMD is compared to the, if I were to coarse-grain using traditional method and do the MD?
So as you said, we fold things in some nanoseconds instead of microseconds, and the simulation is kind of probably 10 times lower. So kind of 100 times faster, 2 order of magnitude faster, yeah, something like that. But it's remember, it's trained on this molecule, so it's not something that you can use until you have the data. So at the moment is that, can we actually build something like this? And the answer is yes. It doesn't fold protein number 13 or 14 or some other protein at the moment.
Okay,
because it's trained on only 12 proteins. So
okay.
it's okay.
Okay.
Okay, so some conclusion here. Deep learning has gone a long way, but in my view, it hasn't been as transformative in drug discovery yet as it was for vision and languages. I mean, it's vision and language has been really mind-blowing. And in drug discovery, you can kind of survive these days without using any deep learning, and it's gonna be fine. Of course, there is the exception of AlphaFold, but this is more for protein structure, not so much for kind of drug discovery. So I expect and I hope that we can get some more disruptive results in the future. Maybe one of the reasons why we cannot do that with deep learning right now is lack of data. No, for vision and languages, you had a lot of data. For this kind of methods, we don't have for this kind of application, we don't have a lot. The new generative models are promising as they can solve some of the initial problem we encounter, in particular for the synthesizability of the molecules. And we're really keen in seeing how this next generation molecular simulation worked out, like where you actually just simulate with an neural potential. And I think there would be, if everything work out well, might actually retire the old molecular mechanics potential, and there will be kind of convergence between different methods. So in these past years, like there was quantum mechanics was one thing, quantum chemistry was one area, and molecular mechanics was one area, and kind of coarse graining, like beads kind of stuff was another area. But now they all use the same methodology. They all use the same learning. No? So we generate data at one level, then we machine learning at the next level, and so on and so forth. So there might be some nice things coming out. And we'll have to wait and see. And this is just kind of thanks to, I try to put it along the way who did what, citing the people. And thanks to everybody for listening.
Perfect. Thank you so much for the great talk. I really enjoyed it. If there's any question, please raise your hand or just come off mute and ask your question. Please.
Hello. Thanks for the very good talk. I have a question on there is a model use transformer, and you said the primary goal using transformer is to speed up. So could you explain more why transformer could be faster?
So why transformer could be faster?
Yeah.
Okay. So before TorchMD-Net, there was ANI, which is faster, but is not equivariant. And there were other equivariant representation which were not fast. So the transformer was a way to have, and there was another one, remember the name, just before us, another method came out which was equivariant. The TorchMD-Net just gave a way more accuracy than that. So it was kind of a nice middle point between accuracy, which was state of the art at the moment we send it out, and speed. But currently, I mean, there are much more accurate methods in the sense that better representation. But I would trade some of that accuracy for speed, to be honest, if I could.
Okay, I thought transformer would be like using more matrix multiplication. So that should be more.
Yeah, but the other one used spherical harmonics, which were even slower.
Okay. Okay, thank you.
Any other question? I have one question about like the generative work that you have been doing. Have you tried to kind of in the you have a reinforcement learning loop at some point? I forgot the name. It was PlayMolecule AI.
PlayMolecule, yeah.
Yeah. Have you tried to use that with like the reward being, for example, the FEP of kind of the ligand with the protein or some sort?
Yeah, that's kind of what the LigDream does. Mm-hmm. So you can have some sort of FEP may be a little bit slow, but you can do other faster way. So score and give a reward and then a loop through. But that's ADIM. That's what we are doing, something like that right now.
Okay, I see. And does that matter? Does the accuracy of the method that the reward matter? Like the accuracy of the noise, like in the reward, does that matter much? Or I haven't seen any bias.
For the scoring function?
Yes.
Well, of course, if it's inaccurate, you would end up with wrong molecule. It's every time you make a molecule, it costs a little, and then it costs a lot of time. So if you have any enrichment there is nice, but if you can avoid make molecule or just make molecule which are more likely to be good, then it's already good. But of course you want scoring function which are good. It's I don't know exactly how the accuracy. So what level of accuracy is required to give some benefits? Do you need a method with good 0.9 correlation, 0.8 correlation, 0.7 correlation? I think 0.7 would, intuitively, I think 0.7 would already be good, but I don't know.
Okay, I think that was exactly my question. Like, what's actually the level of accuracy, like correlation with the two ground truths to actually be useful? Okay, thank you so much.
Okay.
There's a question in the chat. What was the reason for the increased dynamics? I understand model fold in nanosecond instead of microsecond in the ML.
Sorry, what is the reason?
Ricardo, do you mind maybe coming off mute and ask your question?
Sure. Yeah, thanks. Thanks for the nice talk. Yeah, I had a question about the machine learning protein folding at the end. So I was wondering what's the reason for the increased dynamics? I understood you said that the proteins fold on the nanosecond scale instead of microsecond.
Because you flatten you have less degrees of freedom, everything is faster.
So it's like, okay, so it's the kind of normal coarse graining reason, let's say.
Yeah, it's a normal coarse graining reason. The only difference compared to any norm other coarse grain like Martini and so on, the only difference is that we learn a force field which preserves the thermodynamics explicitly.
Right. Okay. Yeah, but maybe that misled me because I thought that
not the dynamics. So the kinetics would be different, but the thermodynamics would be the same.
Yeah, gotcha. Okay, thanks.
Cool. One question from Abik. Have you done any affinity prediction considering water molecule in the pocket with KDeep or something similar?
KDeep predicts affinity.
Yeah.
So
with water molecule in the pocket.
Yeah. So you put the inputs is a protein and a molecule in the pockets, and it predicts the affinity.
Yeah.
But what was the question?
Water
molecule. No. We did something with water molecule, but it did not improve significantly. But again, you have so little data, and then sometimes like you have waters in the crystal structure, sometimes you don't. So you cannot just sometimes put it, sometimes don't put it. So it's kind of noisy, and intuitively it should be better. In practice, it didn't improve much, but so we don't put it at the moment, but it's something that, we tried.
Perfect, thank you. Question from Huang. Could you explain how energy conservation poses a physical constraint for molecular encoding model?
For energy conservation for molecular encoding,
yes.
For KDeep, there's no energy, so there's not any conservation there. But for a force field, you want, for a force field, physics says that in an isolated system, the energy is conserved. So you want forces which have certain properties. And a way to enforce that by construction is to predict an energy instead of predicting the forces. So if you predict an energy and then you compute the forces as a gradient of that energy, then you guarantee that the force field that comes out is a conservative force field. If we were predicting directly the forces, it would be nicer because it would be faster. You don't need to backpropagate on the network, but it would be hard to impose the conservation in the field. So there are some attempts by penalizing the rotor or whatever in the forces in the loss, but is not exact. No, while if you just predict the energy into the gradients, you are guaranteed to have a correct force field in terms of energy conservation. If that answers the question.
I think so. Thank you. One last question for myself. What do you see as the next step to move like this field forward? Is it more modeling, more data? What's missing to unlock, like, to reach this transformative ability of ML in PZ-based and drug discovery?
Well, an easy one would be more data, but it's not easy to get more data. And so I don't know if more models. I mean, I think accuracy, the representational accuracy of the model is already beyond what's necessary from my point of view. We would like faster model, and one way to generate as much data as you like is QM. So that's one way to be transformative, no? So focus on that area, and in that area, you can generate as much data as you like, and hoping that the emergent behavior then is super accurate. That's our hope. But for drug discovery is, there are a lot of tools. It's just they're not like ChatGPT, no? That you're like, wow. It's kind of, maybe they win some challenge like we did, but is the other ones want some other, and maybe like give an example in a challenge that I didn't show, which was a log P prediction, which is one of the characteristics, physical chemistry characteristics of a molecule. We won with XGBoost. We got second in a statistical indistinguishable for first with XGBoost, not with the deep learning method. So yeah, it still produces things which are, but it's like you can train KD on 5,000, 6,000 data points. That's all we have. So it's, if we had 5-6 million, I mean, maybe there would be a difference.